Good morning. Today is Wednesday, June 3rd, 2020. My name is Ricardo Sandoval, and this is the experiment video for week 4, lab 6. This lab is entitled Biochemical Testing for Microbial Identification, Methyl Red, Vosges, Proscour, and Catalase Testing. This is for Los Angeles Pacific University Summer 1 for Biology 225, Microbiology. This lab will be a multi-day lab. This is the start of my experiment, as I do need to incubate specimens of E. coli and Staphylococcus epidermis in a special nutrient broth called MRVP broth for a minimum of 2 days. So this part of the lab, I will go through the process of showing how I incubate the broth. It'll sit for 2 days, and then today is Wednesday, so Friday or Saturday, depending on the growth of the cultures themselves, I will then complete the experiment video. So with that being said, I'm going to don my PPE, flip the camera angle, and go through my supplies, and then we'll start with this part of the laboratory. Okay, so for this first part of the experiment, what I'm going to do again is I'm going to inoculate, if you will, 2 tubes of this MRVP broth with previous cultures of E. coli and Staphylococcus epidermis, or Escherichia coli, that I cultured several weeks ago, and then they'll sit for 2 days. So, and then at that point, I will do a methyl red and a Vosges Proscour test, and at that point, I'll go into more detail about what those tests are and what they do. So I just want to go over my supplies here. So this is my E. coli culture that I just pulled out of my fridge earlier and let it come to room temperature. This is my S. epidermis culture. And then this is one of my MRVP broths. This one is for my S. epidermis growth. And then this one will be for my E. coli. I have 2 pipettes here that are just chilling in alcohol, sterilizing. And then I have here a tea light that I'll use to sterilize the tubes themselves. And then, of course, this is just a quick little slide I made with the information about this lab. All right. So, with that being said, I'm going to go ahead and get started. And then my work surface itself has already been, I already cleaned it before I started with my 10% bleach solution. Okay, so we're going to start with my E. coli here. So let me try to make it a bit easier. Alright, so I'm going to do that. Take off the lid. Run it through the flame here. Just sterilize the tube. I want to still just have that very distinct pungent odor. Okay, so there's that. Now I'm going to do the same thing with my E. coli MRVP broth. And then just flame it. Okay, so that's that. Okay, so my pipettes here. So I'm going to put these over here. Squeeze the alcohol completely out of one. That was just there to sterilize. And then now what I'm going to do here is I'm going to insert this pipette into my E. coli tube, which I have here. I'm going to drop some of this culture and then put 4 drops of it into the MRVP tube that I had labeled earlier. So this is it. So 1, 2, 3, and 4. Okay, the rest of this will go back into the tube. This I'm going to set aside to put in some bleacher in a few minutes. I'm going to flame this again. Okay. Lid back on my broth. Put it to the side. I'm going to flame my E. coli culture tube again. Okay. Lid on, and then we are done with E. coli for now. Alright, so same procedure for the S. epidermis. This is my S. epidermis culture. Just flaming the lid to sterilize. And I'm putting this in a little test tube rack, but it's kind of up here. There we go, that's better so you can see what I'm doing. And then this is my S. epidermis MRVP broth. Okay, that's flamed. Okay, so my pipette here, just squeezing out the alcohol. Okay, just going to set the alcohol to the side for now. And again, I'm going to draw up some S. epidermis. Oops, that's my broth from my culture here. Okay, and again, 4 drops are going to go into the MRVP broth. 1, 2, 3, and 4. The rest are going to go back in here. And this is going to go into some bleach in a minute. Okay, let's flame this again. Okay, lid back on. I'm going to flame my culture vial again. Oops, it's foggy, it's warm in here. It's a very hot summer day in California. Okay, that's flamed. Lid on. Okay, so at this point, that's it. So these will sit for 2 days, and then hopefully have growth in 2 days, and then we'll go from there. Thank you. Good morning. Today is Sunday, June 7th, 2020. My name is Ricardo Sandoval, and this is a continuation of Lab 6, biochemical testing, methyl red, Vogue Proskauer, and catalase testing. This is for week 4 of Biology 225, Microbiology at Los Angeles Pacific University for the summer 1 term. This is just a quick little sign here that I've made summarizing the information I just talked about. So in this part of the exercise, I will be using samples, or cultures I should say, that have been incubating since Wednesday, June 3rd, of E. coli and S. epidermidis to perform initially the methyl red test. So the methyl red test uses this methyl red solution. And what I'm looking for here is basically the results of a process called mixed acid fermentation. And basically, as you know, we can, by the word here, acid, we're looking for, once I do things here, we're looking for results of a red or a pink color, because on the scale or color spectrum of the pH scale, reds are acids versus blues, purples are bases. So basically in mixed acid fermentation, bacteria ferment just by sitting over time, and then they form acids, thereby once the methyl red is added, if the fermentation, the mixed acid fermentation process did take place, it will churn red or pink. So as far as supplies here, of course, I have my methyl red. I have test tubes here. So I have 2 labeled E. coli, and then 2 labeled S. epidermidis. In this exercise, I'll be using 1 of each. 1 E. coli, 1 S. epidermidis for the methyl red test. And then here I have the E. coli culture growth from my E. coli for the methyl red low Prescar broth. So again, it's been sitting since Wednesday, just because I didn't see much in the form of growth. There is some flocculent growth at the bottom, as you can see, but not much turbidity. So hopefully we'll get a good sample here. And then this is for my S. epidermidis. And kind of the same thing. You can see some flocculent growth at the bottom, but not much in the way of turbidity. Yeah, and with that being said, I've already wiped out my work surface here with a 10% bleach solution to sterilize. My test tubes I cleaned with hot water soap, and then alcohol to sterilize. So the first thing I'm going to do here is I'm going to pour half of the E. coli broth into each tube. So half here, and the same thing with the S. epidermidis. So again, this is the E. coli. And then these 2 tubes here are the E. coli tubes. So here we go. So the goal here is to pour in right about the same amounts into each tube. So let's see if it all works out. Well, now that I'm pouring, there is some flocculent growth, and again, this growth does have a bit of a pungent smell to it. So I mean, that may be the result of fermentation. Okay, so that's the E. coli. I'm going to put this lid back on and set it to the side. And then let's see, they do have about, let's just pour a little bit more of this one. We're about the same amount of liquid. Just very interesting that one's very turbid and one's very clear, and it's the same culture, so quite interesting. And then we're going to do the same thing with the S. epidermidis tubes. This is my S. epidermidis culture in the methyl red low Prescar broth. And let's see if we kind of get the same thing here. That we'll get some turbidity once I start pouring. Yeah, same thing here, quite interesting. And again, this also does have a very pungent smell to it, so we'll see if, once I add the methyl red, if I do get that red or pink color, that indicates that mixed acid fermentation did indeed occur. So that's being set over there. About the same amount, more or less. here. Oh, goodness gracious. Okay, that's more or less good. All right, so now that I have half and half of the samples in each tube, I'm going to go ahead and proceed with the actual methyl red test. So, again, I have my methyl red reagents here, solution. Okay. Just going to cut it over a trash can that you can't see. It's kind of off camera here, but forgive me. Okay. So I'm going to put in 7 drops into one E. coli tube and to one S. epidermidis tube. And again, what I'm looking for here is either a red or pink result to see if the mixed acid fermentation did indeed occur. So we're going to start with the S. epidermidis tube. And again, 7 drops. So 1, 2, 3, 4, 5, 6, and 7. And as you can see, it's very dark red. So positive result here. We have mixed acid fermentation in the S. epidermidis. And we're going to do the same thing for the E. coli. 7 drops again. 1, 2, 3, 4, 5, 6, and 7. And the same results here. Very beautiful dark red here. So we do have mixed acid fermentation in both the E. coli and the S. epidermidis. Okay, so I'm just going to take some pictures of these 2 tubes, and then we'll proceed on with the Vogue Proscour test. Okay, so now moving on to the Vogue Proscour test. And in this test, I'm going to use what's called Barrett's reagent A and Barrett's reagent B. So what I'm looking for here is I'm looking to see if the E. coli, which is here, of course, and the Staphylococcus epidermidis go along or use what's called the butanediol pathway and produce what's called acetone. And acetone can be used as an energy source or an external energy source by certain bacteria. Kind of the same principle as mixed acid fermentation, but in using these 2 reagents, the butanediol pathway produces a weaker acid, so the colors themselves wouldn't be as bright as with the methyl red test that I just performed a few minutes ago. Okay, so the first thing I'm going to do here is I'm going to start with my Barrett's A reagent, so Barrett's A, and I'm going to put 12 drops into each tube. So again, I'm going to cut this off screen here, just over my trash can. Okay, so we're going to start by E. coli. One, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. I'm just going to give it a little mix. And then unlike the methyl red test, this is going to take a half an hour before I see anything happening. So this is the Staphylococcus epidermidis. Again, S. epidermidis normally is normal flora, but for patients who have an IV, for example, or a catheter or a chest tube, infections can develop. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. Okay. So that's the reagent A. Give it a little swirl. And then now I'm going to do the reagent B process, and then 4 drops of the reagent B into each tube. I'm gonna cut it off screen here. Scissors over there. One, 2, 3, and 4. One, 2, 3, and 4. Okay, so I'm just going to give these a little swirl here for about 30 seconds just to expose to oxygen and make sure that everything is mixed properly. And then again, these are going to sit for half an hour. So at the end of the half an hour, what I'm hoping to see is a red surface layer. And that would be a positive result that both of these, or one of these, uses that butane-diol pathway and produces acetylene. If I don't see it, then they don't. Okay, so that's the S. epidermidis. And just very interesting here that when I put in the Barrett's reagent A into both cultures here, they both went cloudy, and then with the Barrett's reagent B, they both kind of cleared up. But I will say that from just what I'm looking at, the S. epidermidis, it's much clearer in comparison to the E. coli, which is what's in my hand right now. Okay, so I'll be back in 30 minutes. Okay, so it's been 30 minutes that the E. coli culture from the MRVP broth has sat with the Barrett's reagent A and B, as well as the S. epidermidis culture. So now it's time to see if we have a positive result for the butane-diol pathway and the creation or the use of acetylene. So first off, we're going to start with the S. epidermidis tube. And what I'm looking for is a red surface layer with the culture. So S. epidermidis pretty much looks the same. So I see nothing. So this would be a negative result. And then for my E. coli, oops, you can see a slight redness. Okay, so this is the E. coli. It's developing very slowly, but this I would call kind of, it starts here. It's a very faint red, and this I would call a positive result. So there is acetylene in the E. coli. Okay, so that's the conclusion of the Vogue Proscour test. I'm going to take some pictures of the tube, and then we'll move on to catalase testing. Okay, so now we're going to move on to what's called catalase testing. And the suffix, the ASE in catalase, should let us know that it's an enzyme. And an enzyme, of course, makes things happen quickly. What I'm going to do here is I'm going to use colonies or colony form unit, CFUs, from petri dishes that I've grown of E. coli and the Staphylococcus epidermidis. Those colonies will be placed on a slide using a sterilized inoculation loop, and then I will place 1 drop or so of hydrogen peroxide on them to see if the organism is catalase positive or catalase negative. So hydrogen peroxide, as we know, we use it for an antiseptic. So if you get a cut or a scrape, for example, or if you have a sore in your mouth, we put that on the injury or swish it around our mouth sometimes with a little bit of water. And what we normally get is we get a reaction that the peroxide itself bubbles up. And when it's bubbling up, it's killing bacteria, which is all well and good when we're trying to disinfect. But when it comes to bacteria, in this case, hydrogen peroxide, of course, isn't good to the longevity of a cell or a bacteria. So what catalase does is it protects against the harmful effects of hydrogen peroxide. So the chemical equation, if you will, it's H2O2. So two atoms of hydrogen, 2 atoms of oxygen. So if catalase is present, it separates the hydrogen and the oxygen. So you're left with it with just 2 hydrogens and 2 oxygens, and the oxygen basically goes away as rapid bubbles, so that the oxidative or damaging effects of the hydrogen peroxide don't damage the cell, so to speak. So what I'm looking for here as we go through the process are very rapid bubbles. So I'm just going to go through supplies here quickly. So I have a petri dish here of E. coli and a petri dish of the S. epidermidis. So both of these petri dishes have been sitting in my fridge for quite some time, and they were used for the specimens, if you will, in the methyl red and Vogue Proscour test, and they've developed some condensation and some breaks, so hopefully that won't impact the integrity of the experiment, so we'll see. And then I have 2 slides here, one for E. coli, one for S. epidermidis. I have 2 inoculation loops that are sterilizing in some alcohol. I have a pipette. And then, of course, good old H2O2 or hydrogen peroxide. And again, my work surface has already previously been cleaned, disinfected, sterilized with a 10% bleach solution. All right, so the first thing I'm going to do here is I'm going to grab some colonies from my E. coli and transfer them onto the slide. So pipette here, I'm going to pipette inoculation loop. I'm going to take it out of the alcohol. Just bang it dry. Okay. And now I'm going to open the lid to my E. coli and then grab some colonies, hopefully here. Just dragging it very gently along the surface of the agar. Okay. Lid back on the E. coli, and then I have my E. coli slide, and then I'm just going to transfer the cultures right onto the circle here that I drew with a permanent marker. Okay, and then this is going to go to the side here, so I can soak it in some bleach here in just a second. And I'm going to do the same process with the S. epidermidis. Okay, so I get an inoculation loop out of the alcohol. Okay, so move the E. coli over here, lid off of my S. epidermis petri dish. And then again, I'm just dragging the loop here over the agar just to grab some cultures, some colonies. Okay. Lid back on. And now my slide for the S. epidermidis. I'm just going to place the colonies right within the circle. Okay. So this is going to go to the side to go into bleacher in a minute. So now I'm going to use my pipette. So just to draw up some hydrogen peroxide. Here we go. So I'm going to do 1 drop on the E. coli, which is here, and I'll do my best to kind of show it to you as I do it. And again, we're looking for rapid bubbles. And it's foaming very fervently. So E. coli is catalase positive. Okay. Bubble, bubble, bubble. So again, the H2O2 is now becoming hydrogen by itself and oxygen by itself. So the destruction, if you will, the oxidative properties of the hydrogen peroxide are being protected, the cells being protected, and the O2. That's what's causing the bubbles themselves. Okay, so again, 1 drop on the S. epidermis. Okay. And this, not so much. As you can see, nothing's really happening. So I don't really see bubble formation. It's very, very slow. So we're not having the hydrogen and the oxygen separated here. So the S. epidermidis itself is catalase negative. Okay, so this concludes lab number 6, biochemical testing for microbial identification, methyl red, Vogt-Proskauer, and catalase testing. Thank you.